Hello my dear students, this is Dr. 𝗲𝗵𝗴𝗿𝗺, your biochemistry teacher. Well, in this video of ours, we are going to discuss about cholesterol. So what we are going to discuss in this video, I'll just give you a brief outline. We would be discussing what is cholesterol because you all are familiar with this. Because nowadays in general we hear the level of this person is having more cholesterol, like that. So cholesterol is a very common terminology. Now what we are going to study is how it is produced in the body, because we take it from the diet also. So what happens when cholesterol is synthesized, or what happens when cholesterol is taken in the diet? How it is metabolized in the body, what is the clinical significance, what is the normal concentration, and at what conditions it increases, right? So let's start up with this video, cholesterol. Now as you can see, 𝗲𝗃 is generally we call it as an intestine, okay, and sterol. So we can say that it is the most important animal sterol. Right? Very important. And if we talk about cholesterol, it has a very interesting type of structure. It has a cyclo-pentano-perhydrophenanthrene nucleus. Okay, and basically it is strictly amphipathic in nature. I'll just show you the structure, how this cholesterol looks like. This is the structure of cholesterol. So you can see that it is having a cyclopentanoperhydro, this is phenanthrene nucleus. And the most important point to remember about cholesterol is it is amphipathic in nature. And what we mean by this, that it has both ends. It has a hydrophobic end and a hydrophilic end. Right. Then what are the carriers of cholesterol in the body, which we have already discussed in detail in the metabolism of lipoproteins also. But yes, since we have started discussing about cholesterol, so these are the important points which we need to cover in this also. So if we talk about this, always remember LDL, which is low density lipoprotein, we call it as a bad cholesterol. Why? Because LDL is responsible for carrying this cholesterol from the liver to the extrahepatic tissues. So basically LDL is actually taking cholesterol and depositing it to the extrahepatic tissues. On the contrary, HDL is known as a good cholesterol. Why? Because HDL is responsible for a very important process which is known as reverse cholesterol transport. Now as the word suggests, reverse mane ulta, means HDL does not let cholesterol to be deposited in the extrahepatic tissues. It will bring more and more cholesterol from the extrahepatic tissues and it will deposit them in the liver. And why? Because cholesterol leads to the formation of bile acids. Cholesterol leads to the formation of steroid hormones. And cholesterol is the precursor of vitamin D. And whatever is unabsorbed, that is excreted in feces. So cholesterol plays a very important role. Okay. Now coming over to, first of all, what is the fate of this cholesterol? Means what are the factors which lead to cholesterol and what are the factors which can utilize this cholesterol? So we get cholesterol from the diet, then we get cholesterol from the endogenous source, or it is synthesized in the body. We call it as de novo synthesis. Right. Now what is the fate of this cholesterol? This cholesterol is required, 50% is required for bile acids. So major chunk is actually this cholesterol is required for the bile acids. Then it is required for the synthesis of steroid hormones. And it is synthesized for vitamin D. And unabsorbed cholesterol is excreted in feces. Very important, right? So this is the fate of cholesterol, and we say that it is 150 to 220 milligram percentage or milligram per 100 ml of blood or milligram per dl, right? So this is how cholesterol is metabolized, and this is how we get cholesterol from the diet. Now then, what is the source of cholesterol? So normally if we talk about the source of cholesterol, the richest source of cholesterol is hen's egg, and basically the yolk portion. So hen's egg or the yolk, it is the richest source. Right? Then what is the precursor? Precursor of cholesterol is acetyl coenzyme A. And we know that acetyl coenzyme A has 2 carbon atoms. And cholesterol has 27 carbon atoms. Its structure is 𝗵𝗵𝗶_H. So from 2 carbon atom, we are synthesizing 27 carbon atoms. So this is known as de novo synthesis. One thing I have told you that we get cholesterol from the diet also. Now in what form cholesterol is transported in the body? So I can say the form of cholesterol transport right? It is major chunk of cholesterol moves in the form of ester. And what happens here is, this is a very interesting reaction, I would say. Cholesterol moves in the free form also, and it moves in the esterified form also. And this esterified form is the one which is very, very important. Why important? Because HDL is responsible for reverse cholesterol transport. अब the tragedy with cholesterol is it has an amphipathic structure. So what we do is, if that cholesterol is esterified, the structure will be changed, and from discoidal it will become spherical. Now the question is how it is esterified, what is the name of the enzyme which is required for esterification of cholesterol, and what is the name of the product. Okay. So the reaction is this cholesterol combines with lecithin. What is lecithin? Lecithin, we all know it's a phospholipid. So this cholesterol, when it combines with lecithin, it forms cholestyryl ester and lysolecithin. And the name of the enzyme is 𝗪𝗪𝐶𝑎𝑡. And this 𝗪𝗪𝐶𝑎𝑡 stands for lecithin, cholesterol, acetyltransferase. Right? So cholesterol will combine with a phospholipid in the presence of enzyme 𝗪𝗪𝐶𝑎𝑡 forming cholestyryl ester, and this is the form in which cholesterol is moving, cholestyryl ester. And this is lysolecithin. Okay. And we can say that this cholestyryl ester is the form which can convert discoidal HDL to spherical HDL. right? Because we have to change that amphipathic nature, because initially cholesterol is amphipathic, but we have to convert it into an esterified form so that it can be easily transported. Now the question is, this 𝗏𝗵 enzyme, where at what position this acetylation is occurring? So it occurs at C2. Okay. Now there are two parallel enzymes also. One is 𝗏𝗵, other is 𝗏𝗵. So what happens is 𝗏𝗵 is lecithin, because here the phospholipid is lecithin. 𝗏𝗵 is simple acyl cholesterol acetyltransferase. And what is the basic difference? The basic difference is 𝗏𝗵 is associated with HDL and 𝗏𝗵 is associated in the adipose tissue. Okay. So this is the basic difference. Also we can say that 𝗏𝗵 is mainly associated with HDL right, and 𝗏𝗵 is associated in the adipose tissue, or you can say other peripheral organs apart from adipose tissue. So they will actually convert cholesterol into an ester so that it can be accepted by HDL, and then it will undergo, obviously, because HDL kya karega? HDL will take this cholesterol to the liver, where 50% of cholesterol is converted to form bile acids, right? Now one more, what is the clinical importance of this reaction? Clinical importance. Okay, clinical importance is deficiency of 𝗏𝗵. Deficiency of 𝗏𝗵 will lead to Norham's disease. Very, very important. Deficiency of 𝗏𝗵 will lead to Norham's disease. Now there is one more aspect of this. There are two diseases actually. One is Norham's and the other one is 𝗵𝗵. 𝗵𝗵 disease. So what happens here is Norham's disease is due to complete deficiency of 𝗏𝗵. Complete deficiency of 𝗏𝗵, and 𝗵𝗵 is partial deficiency of 𝗏𝗵. Right. Then second is, in Norham's disease, obviously the concentration of cholesterol will increase because the enzyme required for the conversion of cholesterol to ester is absent. Here also I can say there will be increased concentration of cholesterol. So hypercholesterolemia due to the deficiency of 𝗏𝗵 or 𝗏𝗵 enzyme will be there. Now third important difference is here hemolytic anemia will be there. Here no hemolytic anemia is there. Fourth is corneal opacity will be there. Here no corneal opacity is there because it is just partial deficiency. And then the last difference is Norham's disease can lead to ESRD. ESRD, end stage renal disease. Whereas in this no ESRD is there. Right? So these are the differences, and this is how cholesterol is transported. Then after this, after the transport of cholesterol, then is how we quantify cholesterol, because again it plays a very, very important role in the body. So estimation of cholesterol is equally important in the serum. So identification या estimation of cholesterol is done by Friedwald's equation. And what this Friedwald's equation says? How do we calculate cholesterol? Simply we can say total cholesterol is equal to HDL plus VLDL plus LDL. Now, if we talk about what is HDL, we know it is high density lipoprotein, VLDL is very low density lipoprotein, LDL is low density lipoprotein. And if we have to calculate LDL, so LDL will be total cholesterol minus HDL minus VLDL. And this VLDL is equal to TGs bata 5. TGs over 5. What are TGs? TGs are the triglycerides. So this is how we determine the amount of total cholesterol. Okay. Then coming over to this, this is Friedwald's equation, and we can determine cholesterol by 𝗲𝗵𝗵𝗴 reaction is there. There is 𝗵𝗵𝗵𝗺 reaction. Ghad's reaction, and there is Zach reaction. Right. So this is how we estimate cholesterol. Now coming over to the major thing, how cholesterol is synthesized in the body. Very, very important. Okay. Abhi tak what we studied was, we studied just the nature of cholesterol. What kind of sterol is cholesterol? It is a very, very important animal sterol which is required for the synthesis of bile acids in the body, required for the synthesis of vitamin D, steroid hormones, and then it is excreted in feces. But the most important one, it always moves in esterified form, right? There are different proteins also which are required. Just say if I talk about CTEP, or we call it as CETP also, cholesterol ester transfer protein, because it is just transferring cholesterol ester and taking TAGs from IDL or TGs. So different type of proteins are also there, which we have already discussed in the lipoprotein metabolism. But overall I would say that cholesterol ester is the most suitable transportable form of cholesterol. Now the question is that which enzyme is responsible? The enzyme which is responsible for esterification of cholesterol is LCAT enzyme. And always remember one more thing, ApoA1 always activates LCAT. I'll just mention it here. ApoA1. LCAT is activated by ApoA1. ApoA1, which is the apoprotein for HDL. Okay. Now we are going to study that how this cholesterol is synthesized. So, biosynthesis of cholesterol. Right. Now, again, whenever we talk about the synthesis, we should know what is the site where the process is occurring. So, if we talk about the site of cholesterol biosynthesis is the liver. In the liver, hepatocytes, right? Smooth endoplasmic reticulum, okay? And to some extent, some books may it is given peroxisomes also. And the fraction is cytoplasm. So we can say that the site of synthesis of cholesterol is the cytosolic and the endoplasmic reticulum fraction in the liver. And precursor, just now I told you, what is the precursor? The precursor is acetyl coenzyme A. And always remember the name of the process is It is de novo synthesis. De novo synthesis. Now, the question is how the process takes place. So, whenever we talk about the biosynthesis of cholesterol, we always study it into 5 main steps. So, what are those 5 main steps? Because how many carbon atoms cholesterol is having? It is having 27 carbon atoms. And what is the precursor? Precursor is just having 2 carbon atoms. So, from 2 to 27, what are the main 5 steps involved? So, if we talk about the first step is synthesis of HMG-CoA. Now, what is this HMG-CoA? beta-hydroxy-beta-methyl-gluteryl-CoA. Second is synthesis of mevalonate, right? Then mevalonate will undergo different phosphorylation reactions. Third is formation of squalene. Fourth, we can say formation of lenosterol. And then comes fifth becomes formation of cholesterol. Right. Now the question is that how this HMG-CoA is produced, because this is the first step. Precursor is acetyl coenzyme A. So now see how the synthesis occurs. This acetyl coenzyme A combines with one more molecule of acetyl-CoA, forming acetoacetyl-CoA. Now this acetoacetyl-CoA will combine with one more molecule of acetyl-CoA, and it is going to form HMG-CoA. So this is the first step, formation of HMG-CoA. And if we talk about the enzyme here, enzyme is HMG-CoA synthase. Right? Now this HMG-CoA now is going to form, it will undergo reduction. And whenever it undergoes reduction, the enzyme has to be reductase, and it forms the second important step, mevalonate. And the name of the enzyme here is HMG-CoA reductase. Right. This HMG-CoA synthase is present in the cytoplasm. Right. HMG-CoA reductase is the rate limiting enzyme of cholesterol biosynthesis. So we can say our first step is achieved, formation of HMG-CoA. Then our second step is also achieved, forming mevalonate. Okay, now this mevalonate will undergo 3 phosphorylation reactions, and it is going to form isoprenoid units. Right. So it will be like isoprenoid units 5 into 2. So total you can say it is going to form 10 carbon atoms. Okay, which is also known as geranyl phosphate. Right. This 10 carbon atom will combine with 5 more carbon atoms, forming 15 carbon atom species, which is farnesyl phosphate. Right. And this 15 carbon atom will be again into 2. It is going to form 30 carbon atoms, which is squalene. And this 30 carbon atoms, this is again the third step. This squalene forms lenosterol. which is going to form that again one steroid nucleus. So this is the fourth step, and then comes cholesterol, which is having 27 carbon atoms. So this is how the synthesis of cholesterol takes place. So that is what I have written also. It takes place in 5 steps. And the most important point in which again the students get confused is there is a correlation actually between the synthesis of ketone bodies and cholesterol. Basically HMG-CoA, till the formation of HMG-CoA it is the same. But the basic difference is ketogenesis is taking place in the mitochondrial fraction of liver, whereas cholesterol biosynthesis is taking place in the cytosolic fraction of the liver. So HMG-CoA synthase, which was the rate limiting enzyme in ketogenesis, is present in mitochondria, whereas HMG-CoA synthase, it's not a rate limiting enzyme, but yes, it is present in the cytoplasm. And the rate limiting enzyme of cholesterol biosynthesis is HMG-CoA reductase. Very, very important. Okay. And we know cholesterol synthesis is an anabolic process. Right. So this is how the synthesis of cholesterol takes place. Then we come over to, after synthesis, obviously we come over to the regulation. So when we talk about the regulation, we always focus on the rate limiting enzyme. So this regulation can be, first is by feedback mechanism. We know feedback inhibition is also known as product inhibition. Right. So what happens here is, in feedback inhibition, I will just take the rate limiting enzyme, that's it, because we have to focus only on that. So HMG-CoA is forming mevalonate, and this mevalonate by different steps forming cholesterol. Right, and this is reductase enzyme. So what happens here is that increased concentration of cholesterol will inhibit this with the help of 𝗲𝗵𝗵. So cholesterol, increased concentration of cholesterol is going to inhibit HMG-CoA reductase by 𝗲𝗵𝗵 protein. What is this? It is simply sterol regulating, sterol regulator, you can say element binding protein. Element binding protein. Right. So we can say that cholesterol, whenever there is increased concentration of cholesterol, it has to give instructions to HMG-CoA reductase that no more synthesis of cholesterol is required. We call it as a product inhibition also, or feedback. Then second type of inhibition, which is the most important one, hormonal regulation. Now hormonal regulation, again there are two main antagonistic hormones. And what are the two main antagonistic hormones? One is insulin, the other is glucagon. And with glucagon we can name epinephrine also, because they are the catabolic hormones. Right? Insulin, we know it's an anabolic hormone, so insulin and thyroxine will come here. Now, what happens is, since biosynthesis of cholesterol is an anabolic process, okay, because we are synthesizing cholesterol, that means insulin will always promote this process, and that is what exactly happens. Because if you go back to the history, if you go back on the videos on carbohydrate metabolism, what is the source of this acetyl-CoA enzyme? Whenever there is increased concentration of glucose, it will form acetyl-CoA enzyme, and this acetyl-CoA enzyme will lead to the formation of cholesterol, will lead to the formation of ketone bodies in a normal individual. Okay, so I can say that it is an anabolic process because insulin is promoting it. So insulin is going to activate it, thyroxine is also going to activate it, whereas glucagon is going to inhibit it, and epinephrine also, because they are the catabolic ones. Now the question is, if insulin is activating it, what is the target enzyme? And if we talk about the target enzyme is none other than HMG-CoA reductase. So since it is a rate limiting enzyme, so rate limiting enzyme is HMG-CoA reductase. And this is present in, obviously if it is a regulating enzyme, it has to be present in two forms. So one is the active form and second is the inactive form. Right. Now see, we know if I talk about the it has two forms, and this is the structure of active and inactive. So you can see that it is having OH obviously at the active site, and this is having OP. So I can say that the active form of HMG-CoA reductase is dephosphorylated. And the inactive form is phosphorylated. So always remember, and वैसे भी it is a well-known fact that insulin, being an anabolic hormone, will always accelerate the anabolic reactions. Okay, so I can simply say that insulin will activate HMG-CoA reductase in the active form, which is dephosphorylated. So, if there is high insulin to glucagon ratio, cholesterol biosynthesis will increase. Right? And if we talk about other enzymes also, for example, we can say that first is this HMG-CoA reductase, glycogen synthase, PDH, pyruvate dehydrogenase. These are the enzymes which we have studied. So these are all the anabolic enzymes, and these enzymes are active in their dephosphorylated state. Phosphorylated state. Very important. So insulin and thyroxine are going to promote cholesterol biosynthesis. Okay. And coming over to the antagonistic part, if I talk about glucagon, glucagon will put it in. Since glucagon is inhibiting it, it will inhibit or it will promote the inactive form. And inactive form is phosphorylated. And in general, you also have witnessed in other videos also that glucagon and epinephrine, they have almost the same function. So if I talk about glycogen phosphorylase, glycogen phosphorylase is active in phosphorylated form. So in general, insulin promotes those hormones which are active in their dephosphorylated form, and glucagon promotes those hormones which are active in their phosphorylated form. So this is hormonal regulation of cholesterol biosynthesis. So always remember, insulin and thyroxine are going to increase it, whereas glucagon and epinephrine are going to decrease it. So this is how they are regulated. Then we come over to what is the fate of this again cholesterol, that we have already discussed. So let me just clarify this thing. What is actually happening is, see, we started with the glucose, because I told you when we discussed about the metabolism of carbs, glucose is like a hero in the body. Why hero? Because it is the ultimate root cause of synthesis of so many things. So glucose is always converted to pyruvate. Pyruvate is always converted to acetyl-coenzyme A by a link reaction. So this acetyl-coenzyme A is coming from pyruvate by a link reaction, which we have already studied. And this acetyl-coenzyme A first of all enters obviously TCA cycle. Acetyl-coenzyme A is required for the synthesis of fats, or we can call them as TGs. They are required for the synthesis of cholesterol and for the synthesis of ketone bodies. Right. And if we talk about the fate of cholesterol, acetyl-coenzyme A, since it is forming cholesterol, what is the fate of this cholesterol? This will be converted into the bile acids. So I can simply say that this cholesterol will be converted first of all to bile acids and to the steroid hormones, sex hormones. Okay, and the basic thing is, what are the enzymes? So always remember 7-alpha-hydroxylase enzyme will divert cholesterol to the formation of bile acids, and 27-alpha-hydroxylase will lead to the synthesis of steroid hormones. Because whenever we talk about any biochemical reaction, the enzymes play a very important role. Okay, so whenever hydroxylation is at the 7th position, cholesterol will lead to the formation of bile acids, and whenever hydroxylation is at 27th position, it will lead to the formation of steroid hormones regulated by the genes. So this is again a process regulated by enzyme or the genes. Now we are going to study that how this cholesterol leads to the formation of bile acids. What are these bile acids and what is its significance? Because from day one when I started with the metabolism of lipids, I told you the drawback, or you can say the favorable point about lipids is only one, that these lipids are non-polar. Okay, since they are non-polar, they always require a protein, which is a apoprotein, to be transported. This is the base. So they have altogether a different set. Similarly, if we talk about cholesterol, cholesterol is also a type of miscellaneous lipid. So its structure is amphipathic, but when it is roaming in the body, it is converted into cholesterol ester. Right, and this cholesterol ester again plays a very important role, because once cholesterol ester is produced, then only it is received by HDL. That is why we say that there is a difference in discoidal HDL and the spherical HDL. Okay, similarly cholesterol is required for the bile acids, because again if we talk about the significance of bile acids form bile salts. Right, what is bile juice? Because it is the most important, I would say, the factor in actually in the digestion of the fats. Why? Because again there is a formation of emulsion, because fats are not soluble. So they are very good detergents which leads to the digestion of the fats in the body, which is synthesized in the liver, stored in the gallbladder. Okay. So here again the detail obviously is taught in medicine, so we will be just focusing here how this cholesterol is converted to form bile acids. And what are these bile acids? They are primary, they are secondary, what are the different steps which leads to their synthesis? So cholesterol into bile acids. Now these bile acids are actually of two types, primary bile acids and secondary bile acids. Now what is the example of primary bile acids and the secondary? If we talk about primary, they are cholic acid and 𝗏-deoxycholic acid, right? And if we talk about secondary, deoxycholic acid and lithocholic acid, right? So how these are synthesized? What is their significance? Now since the precursor of this is cholesterol, so what will happen is this cholesterol will undergo hydroxylation at the 7th position, right? That is why the enzyme is 7-alpha-hydroxylase. So what is the reaction? Cholesterol, right? It forms, it undergoes hydroxylation at 7th position, right? And it is going to form 7-hydroxycholesterol, right? And this 7-alpha-hydroxylase, it consists of NADPH, consists of oxygen, vitamin C. These are the three main components of 7-alpha-hydroxylase. Now once this 7-hydroxycholesterol is produced, this in the presence of NADPH and propanol coenzyme A is going to form the primary bile acids. And primary bile acids, just now I told you, this is I already mentioned, cholic acid and 𝗏-deoxycholic acid. So we can say cholic acid and 𝗏-deoxycholic acid, right? So these are the primary bile acids. Now the question is, we can simply say where the process is occurring. So the first step is formation of bile acids, primary, okay? And where their synthesis is occurring? The synthesis occurs in the liver. Again, very important. The synthesis of bile acids occurs in the liver. Now the question is, what is the rate limiting enzyme? The rate limiting enzyme of synthesis is 7-alpha-hydroxylase, right? Then we can say name I have already mentioned, so I am not going to repeat the name, but I will just tell you again, whenever we talk about the synthesis, we always talk about the regulation. So regulation is by this, and the most important point here is that how they are regulated by, I would say, what is the process? So always remember, it is simple feedback inhibition. Okay. So rate limiting enzyme is this, and regulation may we can simply say that if the concentration of bile acids increases, right, they inhibit its own production, which is feedback. So I can simply say that feedback inhibition is there. Then second is, once these bile acids are produced, then a very important process occurs which is known as conjugation, and that conjugation converts these bile acids into salts. Okay. So what happens here is formation of bile salts, which is a very important process in fact. formation of these bile salts. So always remember that these primary bile acids helps in the formation of bile salts. And what are these bile salts? If we talk about the examples, the bile salts they undergo a very important process called conjugation. So I can say that these primary bile acids undergoes conjugation. Where? In the liver. And they are going to form, पहले तो with which amino acids they undergo conjugation? So conjugation के लिए glycine and taurine are the two very, very important amino acids. So what happens is it forms, जैसे if we write about cholic acid and deoxycholic acid, what are the salts produced? Right. So the name of the salts is glycocholate and taurocholate. And second is glycodeoxycholate and taurodeoxycholate. Right. So always remember, first of all, the bile acids are produced. These bile acids will undergo conjugation to form bile salts. Now once the bile salts are produced, what is the significance? The significance is these bile salts, they lower the pKa. So they are actually the ionized salts. And since they are ionized, they are better detergents. Better detergents, or you can say they will facilitate the digestion of the fats, means emulsification of the fats, right? Then the third step, first is you can say formation of bile acids, primary, then formation of bile salts. Then what is the third step? Third is again a very important step. Basically what happens here is there is enterohepatic circulation. So what is this enterohepatic? It is clear from the word itself that entero mane intestines and liver. So what happens in enterohepatic circulation? 95% of bile salts are reabsorbed in the terminal ileum, in the terminal part of the ileum, and via portal vein sent to the liver and reused. Right. So this is enterohepatic circulation ki out of this 95% of bile acids are reabsorbed by the terminal ileum and via portal vein. They are again sent back to the liver where they can be reused again and again. What about the 5%? This 5% is lost in the form of feces. In the form of feces. Right. Then the last step here is formation of secondary bile acids. So always remember, formation of primary bile acids takes place in the liver and formation of secondary bile acids takes place in the intestine. And actually what is happening is these primary bile acids will undergo now deconjugation. Deconjugation and dehydroxylation means both the groups will be removed. Hydroxylation. And this cholic acid will be converted and deoxycholic acid will be converted. right, to the secondary bile acids. Okay, so here lithocholic acid will be produced, which is less soluble and more toxic. More toxic and less soluble. Right? And secondary bile acids will be produced. So you can say that this is how the synthesis of bile acids, bile salts takes place with the help of cholesterol, and this is the main use of cholesterol. So I'll just show you one picture also, which is again a picture relevant to this. You can see this picture, which is actually completely explaining the entire thing. Here you can see that it starts from cholesterol, which we have discussed. It forms 7-hydroxycholesterol. 7-alpha-hydroxylase is the rate limiting enzyme, which is forming 𝗼𝗵𝗔olic acid, cholic acid, deoxycholic acid and lithocholic acid, and that is what we have mentioned here. यहाँ पे चलो I will write here also the lithocholic acid and deoxycholic acid is produced. One second, deoxycholic acid is produced, and you can here see cholic, lithocholic, 𝗼𝗵deoxycholic acid is here, right. So this is formation of secondary and primary bile acids. And one more picture is there which depicts that 7-alpha-hydroxylase and you can say, which I discussed in the starting, 27-hydroxylase is there. So you can see here, if 7-alpha-hydroxylase is there, primary bile acids and secondary bile acids are there, but if 27-hydroxylase is there, it leads to the formation of the steroids. So this is the fate of cholesterol. And if anything wrong happens whenever there is decreased concentration of bile salts or bile acids, or when there is increased concentration of cholesterol, because what happens is, if there is an obstruction in the bile duct, the concentration of cholesterol will increase. So one thing is, this cholesterol will combine with the phospholipid, which is leading to the formation of, which is basically leading to polylithiasis. Right. So there are different things which actually we discuss in medicine in detail. This is just the biochemistry, that how cholesterol is produced in the body and what is the fate, how it is converted into primary and secondary bile acids. So that is all in this video. I hope it is clear to all of you. Thank you so much.